Okay, it's a great honor and pleasure to introduce to you our first keynote speaker, Daphne Koller. Daphne is the founder and CEO of an interesting new company called In Sitro. You can imagine how they came up with that name, but you will tell us later. In Sitro, and where they are trying to do drug development using, all the experience that Daphne has in machine learning and artificial intelligence. But this is only her last gig. She had previous gigs, one of which is she was a professor of computer science at Stanford, where she did fantastic work in applying graphical models to computational biology, among other things. And when she was, at the top of the mountain, she said, well, I'm going to create a new company. And she co-founded Coursera. I think everybody here probably took a course or knows of it. Apparently, there are like more than 30 million people who took courses from Coursera. So really reached everybody in the field. Her CV is plentiful in awards. The one that I like the most is the MacArthur Fellowship that is called the Genius Award, that is only awarded to really people who have great creativity and good ideas. And she's very important, influential, and creative. As I don't say that, right? And Newsweek said that she was amongst the 10 more important people in 2010, 10 magazines, the 100 most influential people in 2012, and Fast Company, the most creative people in 2014. So I don't think I have to say anything else. Thank you, Daphne.
Thank you, Gustavo, for that very generous introduction and for inviting me to speak here, and thank you all for being here today. So today I'm going to tell you about, as Gustavo said, my latest gig, which is in some ways related to a lot of what the Dream Challenge tries to do, but we're taking a somewhat different tack on this idea of trying to synthesize computational methods and specifically machine learning with, in this case, the problem of drug discovery or drug development. So in some ways, this talk tries to put together an in sitro, as you can tell from the name, which is really a combination of in silico and in vitro, tries to put together these two threads of work that I think are each incredibly influential and have advanced considerably over the last few years. And I think the synthesis is going to be incredibly powerful, not just in the context of what we're doing, but just more broadly. So machine learning specifically has advanced incredibly just even in the last 5 years. So if we look back at one problem, which I think is a highlight of the field, which is the problem of image interpretation, the timeline is actually quite striking. So if you go back to, circa 2005 and you would show this image to a computer and you'd ask it, is there a bear in this image? A yes, no problem. It would get it right about 5% of the time. Fast forward 7 years after, you would show this to a computer and it might have come up with something that says, wolf. And now this is no longer a binary classification problem, it's a multi-class classification problem. And it's not quite right, but at least in the right general space, right? Fast forward a couple years beyond that, and this is a real caption for this image. It says, a brown bear is swimming in the water. Now, this is not quite right because the bear isn't actually swimming, but there is a bear and there is water. And furthermore, this is not a one-word label. This is an actual sentence that describes the image. Fast forward 3 years beyond that, and an actual label for this image is two brown bears sitting on the top of rocks. That's pretty amazing. And we now are at a point where when you quantify performance for this image recognition problem, the computer is able to do this problem at superhuman performance, actually better than people can, which I think in the context of 5 years is actually remarkable. And if you'd asked me, in 2012 when I went to Coursera, when I was going to expect this performance, I was maybe 20 years. So this is pretty remarkable. What's enabled this progress, and I think this is a really fundamental point, is really the availability of incredible amounts of data. So the training set for the image captioning model had about 10 million images that were derived from the web, where people sit there and describe their images on Flickr or whatever using these small, short descriptors. And that's what's enabled that, together with simultaneously the availability of effectively infinite amounts of compute data using resources such as the cloud. So those together have enabled the construction of the kinds of models that we're talking about here. And what's really also interesting to understand is that more data really allows rich models to sort of come into their own. So if you look at small data, which is where we were in the image recognition field back in the ImageNet days, for instance, in the early days, 2010, 2012, we would have been there. And at that point, simple models actually outperform the more complex models. But as you increase the amount of data, the performance of the simple models plateaus. And in fact, they all plateau kind of at the same place. And the rich models really continue to get better and better and better as we get more data. And we haven't, I think, seen the ceiling on that quite yet. Now, what is the secret behind the success of these models in this context is the ability to uncover features. So the simple models, by and large, were based on the fact that people really needed to look at the raw data and sort of extract features from it. And it turns out that even in domains like computer vision, where you would think people kind of understand the space because we all look at images all the time, so people would pick out features like edges and corners and circles that look like eyes. And it turns out that people are actually not really good at coming up with features on their own. And what these current models do, because they train end to end, you give them the raw data as input, you give them the target classification task as output, and they construct the features in increasingly complex layers of representation. So they build up things that are edges and then corners and textures and then bigger things like eyes and door handles and so on and so forth, and build up this representation. And that turns out to provide much better performance than manually constructed features. And what's really going on here is that this has enabled the computer to sort of unravel a low-dimensional structure within this very high-dimensional and complex space. If you take the space of images, it's enormous, right? And a 100 by 100 image, which is actually pretty darn small, sits in a dimensional space of 256 to the power of 10,000. So that's a pretty large space. And so this is, and what the computer does is it uncovers the low-dimensional manifold in which these images actually sit and figures out somehow amazingly that these two images that look nothing alike, this yellow van and this blue-white thing, really sit close to each other, whereas and tractors are not quite there, but they're close, and the regular car over there, the sedan, is also kind of close, even though they look nothing alike. Whereas things like animals would sit in a completely different region of the space. And those low-dimensional spaces sort of are able to uncover meaning in interesting ways. So here's one of my favorite examples from a 2015 paper, which is images of men with sunglasses sit in some region of the space. Images of men without sunglasses sit in a different region of the space. There's a distance function that's uncovered so that if you subtract the men with the men without the glasses from the men with the sunglasses and add in images of women without sunglasses, what you get in the space is women with sunglasses, which is like, I don't know, mind-blowing. And if you think this might work in pixel space, the answer is it doesn't. You get garbage. And you get that not just in images, you get that in other spaces. So here's another one of my favorite examples. And this is in the space of, this is the word tvec, which captures this in the context, which tries to capture semantic meaning of words, simply by using nothing except the context in which words are used in very large amounts of text. And it turns out that it discovers that man is to woman as king is to queen, or walking is to walked as swimming is to swam, or Spain is to Madrid as Germany is to Berlin. All just using raw data. So that's all great, and that progress has been remarkable and is continuing in a wide variety of domains, but it hasn't made as much of an impact on biology quite yet. And so let me talk a little bit about the second thread that I'd like to put together here, which is the thread of biology. Biology too has made, and specifically medicine, has made tremendous strides in the last 50 years. I mean, starting from early days of polio vaccines, then the 1990s, we had biologics, which transformed our ability to drug with great specificity key targets that have provided us, for instance, with a whole new generation of anti-cancer drugs and anti-inflammatories, cancer immunotherapy just in the last few years, and of course the recent Nobel Prize for that. The ability to take genetic data and really at precision scalpel level understand the genetic mutations that underlie diseases such as cystic fibrosis and provide targeted therapies for people with very specific mutations. And then, as we're now seeing, the new generation of cell therapy starting to emerge. So that's all great. So awesome, except not so awesome. So some of you might have seen this graph. It's a graph called Eroom's law. Eroom is the inversion of Moore's law. And what you see here is the number of drugs discovered per $1 billion of the R&D spending. You'll notice that the graph is on a log scale, and you'll see that there is this downward line which doesn't correspond to a good trend of, the cost of computing going down exponentially, but rather the cost of drug discovery going up exponentially, or the number of drugs per $1 billion going down exponentially. This is not a good graph, right? And if you look at why that is, in 1970s, it cost a few $100 million to discover a drug, and now in the 2000s, it costs $2.5 billion to discover a drug. Now, this is not because the cost of the investigation of any single molecule has gone up in this ridiculous way. In fact, that cost has actually gone down as new technologies have been brought into bear. What's actually gone up is the number of failures. So what we see is that the number of drugs, if 10,000 compounds go into step one of drug discovery, at each stage of the funnel, that number goes down and down and down. So that one compound that actually makes it has to carry the weight of all of the failures for which we paid those costs. And that's what's led to that $2.5 billion number. Now, if you step back and think about this, this is actually a prediction problem. That is, if we could look at those drugs at the beginning and say, these are the ones that are more likely to fail, and these are the ones that are less likely to fail, then we could toss out the ones that are more likely to fail and have fewer failures to carry through the rest of the process. Now, we're never going to get this perfectly because biology is really, really hard. But if we could reduce that number of failures, that would hopefully help us do better. And so if this is a prediction problem, the question is, machine learning is good at prediction. Can we help make this better? So what we're trying to do at In Sitro is basically get at that question. So try and create a new paradigm for drug development that uses high quality data and data-driven models to design novel, safe, and effective therapies that help more people faster and at a lower cost. So machine learning has been applied to biology in a number of ways. There's a number of different really interesting applications, some of which have been presented at this conference. I'm going to talk about this one because it's one of my favorites. Actually, sorry, the animation's a little wrong. So this is actually one of the last papers that I wrote before, the one on the left, before I left Stanford in 2011. So this is joint work with one of my students, Andy Beck, who's an MD, PhD pathologist. And Andy basically wrote what I think is the first digital pathology paper that applied machine learning to these H&E stained images. And we took these breast cancer tumor images, the same one that pathologists have been looking at for literally 100 years and doing cancer grading. And instead of just using the standard grading mechanisms, we we quantitated hundreds of different features. This was pre-deep learning, so this was manually constructed features, but it was all sorts of weird stuff that people had never really looked at. Put this into a machine learning classifier, really simple, it was logistic regression with ElasticNet, and tried to predict 5-year survival, because that was the label that we had. And it turned out that this was able to classify or predict 5-year survival much better than a competent pathologist. So that was one thing, but to me, what was far more interesting about this paper is not just the performance that it got, but actually the fact that it was able to do that by using all those features that pathologists had never looked at. And specifically, the features that it used were really weird ones, like the average distance between a tumor cell and the closest stromal cell. Now, if you step back and ask what it was really sort of anchoring on, it was anchoring on characteristics of the stromal tissue that surrounds the cancer. And now you would say, well, obviously that makes sense because it's the characteristics of the tumor microenvironment, which we all know is important. But this was 2011. The computer didn't know anything about the tumor microenvironment. We didn't either. So you could say that in some ways what the computer was doing was among the earliest harbingers of the importance of the tumor microenvironment for cancer prognosis. So this, to me, is a really nice example of the value of data-driven discovery. So fast forwarding, I just want to take you through this progression because I think it's a really nice one. This was 2011. In 2018, there's a bunch of papers. I just picked one almost at random because they all achieve, at this point, performance that using deep learning methods that is much better, not just than your average pathologist, but than an expert pathologist. And you can see the Kaplan-Meier curves being really nicely separated between the computer on the left and the pathologist on the right. But what I think is really more interesting is the final step in this progression. This is the paper that came out, I think, 2 months ago, that used deep learning to do not just better than a pathologist, but something that pathologists had never been done before, cannot do and has never done before, which is look at a tumor sample image and identify not just whether this is cancer or what the prognosis is going to be, but what specific mutation gave rise to that cancer. Is this a P53 cancer or an EGFR cancer? No pathologist can do this. So this, I think, is a really nice example of machine learning in medicine. But what does that have to do then with drug development? How do we apply this to drug development? So remember what I kept telling you at the beginning, and is also the case for the pathology example that I just gave you. The driver of success here is having really large amounts of data. And we're now in a world where large amounts of data are finally becoming available in the context of medicines. And so let's talk a little bit about where some of those really large data sets are to be found. One of those is the number of human genomes that we're now able to get access to. So this is a graph that looks at the number of human genomes sequenced starting from 2000. This is from a paper called Big Data Astronomical or Genomical. It came out in PLOS in 2015. And it starts out with the first human genome on the left in 2001 and then goes up. Note that this is a log scale again. And so what's interesting about this line is that not only is it growing exponentially, but it's growing twice as fast as Moore's law. And so depending on whether you believe that this line is going to progress in historical trends or slow down to the Moore's law line, the number of human genomes sequenced by 2025 is estimated to be between 100 million and 2 billion. That's a lot of genomes, okay? Now, genomes by themselves are really interesting, and consortia like EXAC and NOMAD have actually extracted some interesting things just from the genomes themselves. But what's even more interesting to me is the fact that these are now being combined with a very rich set of human phenotypes. So resources like the UK Biobank, which is just an incredible data set, measure not only the genetics of individual humans, but also various molecular biomarkers, human physiology, lifestyle and histories, various cognitive and functional testing, whole body and brain imaging for 50,000, soon to be 100,000 people. There's a whole slew of data that can then be tied to the genetics to really come up with interesting genotype-phenotype connections. And sure enough, by resources like the UK Biobank, as well as many others, the number of those associations between genotype and phenotype using technologies like GWAS has been increasing dramatically over the past few years, and there's now a lot of places where we have an understanding that these are variants that are tied to a particular phenotype. Great. Except that it's really hard to interpret these if you want to actually make use of that of those conclusions. So this is a graph that shows the effect size of variants as they relate to different traits. How much effect does a variant have on a particular phenotypic trait? And so if you look at this, what's really important to note is that all of these graphs are centered smack around 0. And that they all kind of tail off at around 0.03. So we're talking about an effect size of 0.03 being where most of these variants sit. And so do these variants matter? We don't know. Maybe they matter a lot. Maybe they don't matter. Maybe they matter a lot. It's just that in human genetics, because of negative selection against variants that really make a difference, it just doesn't show up as being statistically significant. But if we could really perturb it, it would actually make a difference. And we just can't tell from the genetics alone. What we do know, this is from a very recent paper that came out, is that even though individual variants don't make a big difference, together they do make potentially quite a big difference. So this is from a paper that came out this year, Kara et al., and shows that if you take all of these little variants, the tiny, tiny ones, and put them together in a polygenic risk score, which at the moment is still just a linear scoring function because we don't have as many human individuals as we need to fit something that's more complex than a linear model. What you end up with is when you start looking at the tails of that distribution of polygenic risk scores, you end up with odds ratios of disease that are on the order of as many as 10 to 20 in terms of the odds ratio. And that's about as large as you would have from a truly penetrant Mendelian variation. And so together, all these little variants add up in significant ways. So that's great, but then how do we use that data to really get at the problem that we really care about, which is the effect of human genotype on phenotype, so that we know which genes make a difference, so that we know which genes to drug, because that's fundamentally what we're looking for. So if you look at this, this is a genotype to phenotype prediction problem. And well, if we could apply clever machine learning, maybe we'd really be able to tease out that relationship. But the problem is that that space is just really, really high dimensional. So even in the recent NOMAD consortium, there's, and just looking at the exomes, there's 17 million variants, okay? And that's at the exomes, and we know that of the variation that affects human phenotype, 90% of the variation actually sits in the regulatory regions. So this is a gross underestimate of the number of variants that make a difference. So this machine learning problem is really hard because it's a truly high-dimensional space, and there's just not enough individuals in the world, certainly not enough that have been sequenced, to really get at this relationship. So our premise, what we're trying to look at, is how molecular data could supplement this very limited, relatively limited genetic data. And so the idea is, can we add high-content biological data to really inform this genotype to phenotype mapping? And this is where, again, in just the last 5 years, there's been an incredible advancement across multiple fields that provide us with the ability to create a perfect storm of data production in biology. And those sit in 3 major areas. The ability to construct cellular systems that are much more relevant to human biology than the type of multiply passaged cancer cell lines that have been the workhorse of most of data production for the last few years. We're now able to take a stem cell. We're able to take a fibroblast from a human, de-differentiate it into an induced pluripotent stem cell, and then differentiate it into a number of other relevant lineages, whether it's a cardiomyocyte or a hepatocyte or a neuron or microglia. All of these are differentiable from starting with a single skin sample from a human patient. You're able to perturb those systems, not using small hairpin RNAs where the off-target effects are often an order of magnitude larger than the on-target effects, but using multiple variants of CRISPR that act with much greater precision to enact the perturbation that we want. And finally, the ability to take those output and measure not just growth, but actually a range of high-content phenotypes that include single-cell RNA-seq, that include metabolomics and proteomics, that include imaging that provides subcellular localization of molecules within the cell. That is an incredible wealth of information that allows us to really probe at the effect of genetic perturbations on phenotype. And this is what one of those images looks like. This is actually the first data out of our lab. This is a cellular staining of a bunch of cells, and you can really see cellular substructures here. And this is actually, you can get even more high resolution than this. So what we can now do is we can put all these technologies together and really create, and you can't actually see any of this because the screen's not working. That's exciting. Okay. It's very, very dim. Never mind.
Is there a way we can turn off the lights, maybe?
That's okay.
No?
It's
Okay.
just, it's fine. You can see lots of images of cells dividing and lots of cool, anyway, it doesn't matter. So what we're really building is a data factory. It's something that takes all of these techniques together, along with automation and microfluidics, and allows you to really do a biological experiment at the scale that will allow us to provide the data that drives machine learning algorithms. Not the few hundred samples that we all are used to working with, and not a few thousand samples, but hundreds of thousands or millions of samples, so that we can get to the scale that machine learning models can actually make a difference, and we are at that ever-increasing curve that I showed you that we're at for images. And so by creating a very tight loop between machine learning on the one hand and data production on the other, I think we're going to be able to drive machine learning models that can actually solve really meaningful and interesting problems in the drug development process, including most notably the understanding of how genetic perturbations can drive phenotype. And so I'm going to take a big step back. This is the last couple of slides in this, and really just think about this from a historical perspective. Science proceeds in epochs. The late 1800s, if you think about this, was the epoch of chemistry. It's where we moved away from alchemy and trying to turn lead into gold and understood the nature of the periodic table. In the 1900s, that was the epoch of physics. It's where we understood the connection between matter and energy, and we split the atom. 1950s was the epoch of computing, which is what brought many of us here to this room today. And then 1990s, there was an interesting bifurcation. The epoch of data, which emerged directly from computing, and drove a lot of the advancements that we know of in machine learning. And then at the same time, there was an epoch of what I would call measuring biology, which sits directly on the advancements of both chemistry and physics, and led to the development of things like high-resolution microscopes and the sequencers that we use to sequence, and many, many other tools that are able to quantitate biology at scale. These were two threads that have largely proceeded in parallel with relatively little overlap. But now I think what we're starting to enter into is the era of biological data science as being one of the larger disciplines that will emerge in the 2020s and beyond. It's putting together those last two threads that I just talked about into something that's going to be considerably larger than the sum of the parts and will allow us to quantitate and understand and fundamentally at the end engineer biological systems in a completely different way. And I think that's a really exciting field for all of us to be in. Thank you very much.
So now we're going to have the fire chat between Gustavo and Daphne. continuing this.
So let's take a seat. Fascinating talk. So before I go ahead, and we were thinking of chatting a little bit about things that maybe were not said formally in the presentation, but rather in a informal talk. But rather than me starting, I would like everybody to ask questions, as one normally gets inspired by such a talk. So questions? Can you guys go and there is one over there as well. Go ahead.
Hi. So I was wondering if you could describe some specific examples of how you see that second edge you showed in that figure from going from ML to the biological data factory.
So, is this working?
Use this.
Okay. Okay. Is this working? Okay. I mean, I think there's a number of different ways, actually, that we see this working. One of them, whoa.
Fireside.
Yes. Well, given how cold it is outside, this is good. So, first of all, is just by putting the right people in the room together, because so much of, I think, data production in many places happens without any discussion with the data scientists who are going to then analyze the data at the end. And so the experimental design is often one that doesn't lend itself necessarily to the application of good machine learning techniques. And I think that, to me, is one of the most obvious places where one can make a difference, is just in the experimental design. Then I think the next phase often happens when you actually start thinking about what to measure. For instance, we sometimes find that, biologists can measure a lot of things, but they can't measure all of them at once because there is a limit to the number of photons that you can get from a sample. Which ones should you measure that are going to be the most informative about the task that you actually care about? That makes a difference. You can construct the model by thinking about what matters in the biological system and to the biological problem. And so the fact that people from these two disciplines are actually working together hand in hand provides an incredible opportunity for just making the whole pipeline better all along the way. And I think that, to me, one of the big limitations that I find in most organizations is that they're very siloed, and that you have just people from the different disciplines working entirely separately. And what we're trying to build is really a unified culture where it's a single company and everything is done from the problem selection through the experimental design, through the what to measure, how to model, how to interpret the results, and then what experiment should you perform next, is something that's done entirely in collaboration.
Hi, great talk.
Thank you.
I have so many questions, but I'll limit myself to one. Going back to the funnel. So the problem with the funnel isn't necessarily entirely a prediction problem, maybe you'll agree, is that, and if you maybe talk to medicinal chemists, they can probably, draw up on a whiteboard a lot of compounds that probably will be effective against a specific target. The problem sometimes is just that they'll also just kill you, or they're totally toxic, or you can't deliver this molecule somewhere. So is part of your approach also that there are these other tasks that have been disconnected from the process of stuffing compounds into the funnel at the top, that you now need to sort of consider to increase your chances of success, to getting one out at the bottom?
Yeah, so you're absolutely right, except I will quibble with your first statement, which is that it's easy for a medicinal chemist to come up with multiple compounds that will actually hit the target.
Sure.
But putting that aside, I completely agree with you. A lot of the failures are that, a compound, you put a compound in, it has distribution issues, it has PK issues, it has toxicity issues. All of these are also prediction problems, and all of these can also be potentially something you can train to with a predictive model. So I think, sorry, go ahead.
And very much agreed. And I have a follow-on question to that observation, very small.
Very little question.
Very little one, which is that a lot of the, I think, nice advances in the deep learning successes in vision or caption generation also are sort of accompanied by some concept of certainty or uncertainty. And in our case specifically, we've got a real problem with, like, we have too little data historically. We have a lot of uncertainty. Are your models also sort of taking that into consideration too?
Well, I think the fundamental problem of not enough data is exactly what we're trying to solve. So the whole premise of what we're going into this with is that these are really important and expensive problems. And you can come at them from the perspective of, let's see what data is already out there, has already been generated, and try and glean something from it that has passable performance. Or you can come at it from the other side and say, if this is a really important problem that we actually care to solve, then let's generate enough data so that you can solve it with high accuracy. Because even if you pay $1 million to construct a data set on, say, toxicity or PK or whatever, that $1 million is going to more than pay for itself in the avoidance of failures in the much more expensive stage of what happens when you actually put this into a human. And so it's inverting the problem. Instead of asking, what can we do with the existing data, it's asking, what do we want to do, and then let's generate the data so that we can do it well.
Hi, I have a rather, thanks for the exciting talk first. I have a rather technical question. So we talk about different data sets, it's really exciting, imaging data, single cell data, but how do we reconcile the fact that like even for, very simple linear model, we really struggle to tear out those correlations between the data, for example, the ethnic background, population structure. But I would imagine all those data sets, when we try to incorporate more, there's more and more, complex structure and correlations, but how do we deal with that?
I think complex structure and correlations also exist in all of the other problems where machine learning has made progress in the last few years. I mean, think about how much correlation structure there exists in images. The way we've been able to get past that is by having enough data and the sufficiently rigorous training regime with hyperparameter fitting and so on and so forth, so that you're able to tease out the signal from the noise. I completely agree with you that as long as we're dealing with data sets of size 100, that correlation structure is not something we can possibly ever disentangle, which is why we need lots and lots of data, because that's what machine learning success ultimately relies on.
Thanks.
There's one question over there.
Thanks for the great talk. And so I have a question related to the actual machine learning techniques. So in terms of all the available machine learning techniques used for the prediction in for biological data, so may I have your comments on current available existing data techniques, and what can we expect in the next, let's say, 10 years also? Thanks.
So I'm sorry, I'm not sure I fully understand the question. The question is, what are the techniques that we can expect in the next 10 years?
Yes, this is my second question. The first question is, do you have any comments on current available techniques? Let's say artificial neural networks, deep learning, and support vector machines, so on and so forth. Thanks.
Well, I think that I tend to try and avoid the silver bullet mindset, which is that there is one technique that is good for all problems. And I think there are problems that, because of the quality or the availability of the data or its structure, lend themselves to one approach versus the other. So I don't know if I have some unique insight to offer. I think that what's important is to really look at the problem and understand enough about this, its properties, so that one can see which model class is more appropriate for that data set. So I'm sorry, I don't have a more insightful answer.
We'll have two more questions, yours and then mine.
So I have a question about your new company, In Citro. Could you maybe give us one specific example of disease or biological system or drug or something that you're planning to work on, or you've worked on? No.
See, it's a short answer. No, we're not talking about that publicly right now.
Okay.
It's very new.
Okay, so minding that we are a little bit late with respect to the session, I would like to ask two final questions, and then we can continue the conversation with coffee, without the fireside. But so the first question is, will you give us some data for dream challenges when you have them? And you don't have to answer that. The more serious question is, missing in your grand scheme of what you are trying to do, I think, is more mechanistic and causal thinking that is, I think, a great complement to something which is more data-driven, right? So how are you going to incorporate the fact that we think in terms of models and causality?
So I'd like to disentangle a mechanism from cause in the response to your question. So when you think about causality is ultimately, as Judah Pearl has taught us, is when you perturb something, what effect does that have? So it's the effect of a perturbation on the outcome. And because we're doing perturbation experiments, or can now do perturbation experiments, causality is something that will actually emerge because you can do the experiment. What is, and in some sense, causality also emerges as we start to think about, when we think about the human GWAS data, because that too, nature has done the experiment of perturbing a gene, and we see the effect on phenotype. What that doesn't give you is mechanism. So causality and mechanism in this respect are distinct. And in fact, one of the challenges of GWAS and the challenge of CRISPR and similar technologies is that you don't actually know why it is that when I perturb, or when nature perturbs this gene, it has a particular effect on the phenotype. And I agree that's a really important problem that scientists that science ought to be looking at. I think the question that I would ask is, to what extent is that necessary or sufficient for drug development? That is, understanding cause and effect is absolutely necessary for drug development. Understanding the entirety of the why is, there's a lot of drugs today. I would think the number I've heard is around 15% of drugs that we don't understand the mechanism, and yet they work. And then there were a lot of drugs where we thought we understood the mechanism, and we put them in the human, and they didn't work. So I would argue that it's a good thing to have, but it's neither necessary or sufficient, and maybe we can leave that to scientists while we put more drugs in people.
And that's the last word. Thank you very much.